Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties of the enzyme in the course Enzyme Science and Technology. And in the current module, we are discussing about the different types of host, what is available for the protein production, and how you can be able to utilize these hosts for the protein production. And so far what we have discussed about the prokaryotic expression system. So where we have discussed about how you can be able to transform the bacterial cells with the help of the different types of vectors. And then we have also discussed about the how you can be able to induce those cells, and then you can collect, break opens, and then how you can be able to analyze the protein production with the help of the SDS page. In the subsequent to that, in the previous lecture, we have discussed about the yeast and as well as the insect cell line as an expression system. And we have discussed about the protocols and the procedures what you have to follow to express the protein into some of these expression system. Now in today's lecture, we are going to discuss about the mammalian system as an expression system. So let's start the lecture today. So mammalian expression system, as the name suggests, it is actually going to utilize the different types of mammalian cells for the protein production. So similar to the other expression system, the protein production in the mammalian expression system can be achieved with either from a vector present in the extra chromosomal DNA, which is going to be a transient expression, or the sequence is integrated into the genome through the homologous recombinations to establish the permanent cell lines. The expression from the transient or the permanent cell line can be from the constituted or the inducible promoter. Just like as we have discussed about the yeast expression system, here also you can have the choice of either you want the constitutive expression or the inducible expressions. Irrespective of the expression mode in the mammalian system, the different basic topics need to produce the protein are as follows. In the step 1, you are going to clone the foreign gene in the mammalian expression vector. In the step 2, you are going to transfect the cell lines with a recombinant construct. In the step 3, you are going to screen and select the transformed cells. In the step 4, you are going to culture the transfected cells. And in the step 5, you are going to do the protein production. If it is a inducible expression system, you are going to add the inducer. If it is a constitutive expression system, then you are going to culture the cells for cell generation, and the expression is going to be correlated to the amount of protein what you are going to express. So these are some of the cell lines what you're going to use. You can use the MCF7, HT1080, EH927, and you can use the macrophage cell line GA774. And all of these cell lines have their own origins, so you can actually be able to use the cells as per the origin of the protein as well. For example, if you are trying to express an enzyme which is mostly been found in the kidney, then in that case you should use the CB1 cells. Okay. Similarly, you can use the cells like fibroblast cells for protein production or the enzyme production, where you can use the COS7 or NIH3T3 cells. Then you can use the J774 A.1, which are actually the macrophage cell lines. And then you can use the CHO K1, which is actually Ovidian cell lines, and HeLa, BHK1, and HEK293. And HEK293 is a kidney cells. So depending on the source of the enzyme, you can actually be able to choose the different options. And sometimes the rate of these cells and as well as the maintenance is also another parameter what you can also be keep into the considerations. Mammalian expression system, you can actually have the two options. Either you can have the transient expressions or you can have the permanent expressions. In the transient expression, the expression is high, but for the short period of time, because the vector is not going to integrate the DNA into the genome, so it is actually going to be remain as the extrachromosomal DNA. So the cells transfected with DNA expresses the protein until the 72-hour post-transfections. Transient expression system is used for screening the cDNA libraries, isolation of a particular cDNA clone expressing surface antigen, and to test the applicability of the recombinant construct going to use for the permanent expression. So transient expression is mostly being used to test that your construct is correct or not. Then you can have the permanent expression. The permanent expression of a gene is possible if it will be integrated into the chromosomal DNA. The most crucial step to establish a permanent expression system for a gene is the frequency of integration events rather than the number of gene uptake. In simpler words, the permanent transfection depends on the recombination frequency instead of the transfection efficiency. So transient expression system. So the expression is very high, but for the short period of time, the cells transfected with the DNA expresses protein until the 72-hour post-transfection. So how you're going to do the transient transfections? You can have the multiple steps. So there are multiple steps required for the transiently express a protein in COS7 cell line. So I have taken an example of the COS7 cell. The steps are as follows. In the step 1, you are going to clone the foreign DNA into an appropriate mammalian expression vector to obtain the recombinant DNA. Transfection efficiency is maximum for a supercoiled DNA. So you can purify the recombinant DNA by a mini prep kit to prepare the high quality supercoiled DNA. And in the step 1, you're going to do the delivery of the, you're going to do the cloning, and as well as you're going to do the transfection. So in the step 1, you're going to first plate the uniform cells, right? You can plate 10 to the power 7 cells, and you can actually be able to do the transfection. And in the step 2, you're going to do the screening. So you're going to do the transfect the cells. So in the step 1, you are going to do the screening of the cells with the help of the appropriate antibiotics. And when you do so, you're going to have the death of the most of the cells except the transfected cells, right? So for example, if you use the puromycin or other antibiotics which are going to kill the non-transfected cell, then you can actually be able to select these cells with the help of the screen the cells with the help of the antibiotics. Uh, then the steps 4, you can have the two choices. Either if the expressing cell surface or intracellular protein. So you can have two choices. Either the protein is expressing on the cell surface, or it is going to be an intracellular protein. So transfected cells are allowed to express the protein for another 72 hours. You remove the media, wash the cells with PBS, and detect the presence of protein on the cell surface or in the cell lysate by an activity assay or by the Western blotting. That anyway we are going to discuss in the later part of this lecture, how you can be able to use the Western blotting to detect the proteins. But what you're going to do is you're going to collect these cells by the centrifugation, so that will give you the two fractions. Either you're going to have the cell fractions or the supernatant. So if you have put the cells under the secretory pathway, it is going to be in the secretory into the supernatant, and if it is be present in the either with the cell membrane or with the intracellular protein, it is going to be present on the cell surface. So in that case, you lyse the cells with the help of the appropriate lysis solutions, and that will actually going to release the content. So it is going to give you two fractions. It is going to give you the pellet fractions, and it is going to give you the cytosolic fractions. And cytosolic fractions is going to be it's going to be the soluble proteins. The pellet fraction is going to be the cells which are actually going to be expressed on the cell surface, because that is going to be membrane fractions. So in the steps, if you are expressing the secretory proteins, wash the transfected cells with PBS and add the serum-free media. It allows you to secrete the proteins for next 72 hours, harvest the media, remove the dead cells and debris by the centrifugations, and filter the media with a 0.45 micron syringe filter before storage. Detect the presence of the protein in media either by the activity assay or by the Western blotting. The second option is, so you can also be able to do the permanent expressions. So permanent expression is the permanent expression of a gene is possible if it will be integrated into the chromosomal DNA, and the permanent transfection also has the multiple steps which you have to follow. So in the Permanent expression, you have the different steps. You can have the clone the foreign DNA into the appropriate mammalian expression factor to obtain the recombinant DNA. Transfection efficiency is maximum for a supercoiled DNA, so you can purify the recombinant DNA by a mini prep kit to prepare the high quality supercoiled DNA. Okay, because the transfection efficiency will actually increases the chances of the DNA to be get integrated into the genome. You seed the cells in a DMEM media supplemented with 10% FBS at 20% confluency in a 100 dish. I'm sure many of you don't know that what is mean by 20% confluency. So 20% confluency means the number of cells which are actually going to cover the 20% area of that particular dish. Then you can do the transfection. So transfect the cells with a recombinant DNA using a transfection reagents. You can use the lipofectamine, you can use the BE-based transfections, and you can do the other methods what we have discussed in the previous lecture. Then in the step 4, you're going to do the selection of the transfected cells. You can have multiple options of selecting the transfected cells. You can take the small aliquot of the cell and test the expression of the foreign protein with the Western blotting. In addition, the integration of the genome into integration of the gene into the genome can be checked by performing a Southern blotting with a radioactive probe derived from the gene of interest. So I have not discussed about the Southern blotting, but if you are interested, you can actually be able to see one of my another MOOCs course where I have discussed. So this is called as experimental biotechnology, and if you go under the experimental biotechnology, the I think the module 11 is actually going to be discussed or going to be deal with the southern blotting. So you can actually be able to understand the different steps and as well as the technical details how you can be able to perform the southern blotting to check the presence of gene. Basically what you're doing is you're taking the genome, digesting it into the different with the help of the suitable restriction enzyme, you're resolving it onto a agarose gel, then transferring those DNA to the into a nitrocellulose membrane, and then you are probing that with the small probe, radio labeled probe. Then that probe is going to be the corresponding sequences what you are going to related to that particular gene what you have cloned. And using this, you can be able to say whether the gene of your gene what you have cloned is actually being present in the genome or not. So that you can actually be able to follow if you are interested to read more about the Southern blotting. This is what we are going to do. So we are going to do first first you're going to do a plating of the cells. Then you're going to do transfection with the help of the suitable transfection reagents. Once you got the transfection, you're going to do the antibiotic screening. You can use the puromycin, valinomycin. You have a lot of options of different types of antibiotics what you can use. And then you are going to do the selection of the transfected cells. So in the selection of the transfected cell, you are going to do the two options. So 48 hours after transfection, split the transfected cell in a selection media containing antibiotic and allow it to grow for another 4 days. When you do that, it is actually going to kill most of the non-transfected cell, but it is going to maintain the transfected, going to allow the proliferation of the transfected cells. Then you are going to do is you're gently wash the cells with PBS and observe the discrete colonies. So what you see is you're going to see a discrete colonies of the cell, right? So these cells are not, these are transfected cells, and they are not going to be die with the help of even with the antibiotic, because the vector is providing them the resistance. Delineate the boundary of each colony with a marker from the back side of the plate. So what you're going to do is you're going to monitor your and see the plate is you can be able to just put a marker, and you back from the back side of the plate you're going to say, okay, this is the colony I have, right? So from the back side of the plate you can just put the marker. Then what you're going to do is you're going to put the remove the media and put the cloning rings into the each colony. So for example, if you have, this is the colony you have marked actually, so you can just put a cloning ring actually. Cloning ring is actually a small tube like structure, so it's actually going to be like this, okay, small, okay, and this side is going to have a, the glue actually. So when you remove it and you keep it on this, it is actually going to remain sticked, and then you can actually be able to do whatever you like to do with this. It's going to be get isolated from the other colony. For example, this is also another colony, you can put another colony reactions. So whatever you want to do with this will not going to affect this, because it's going to be sealed from the bottom. So wash the colony with the PBS and add 100 microliter of trypsin EDTA. So in this one, you are going to add the trypsin EDTA. So what will happen? This colony is going to be detached from the plate surface, and that you can be able to collect. So wash the colony with PBS and transfer it into a 24 well dish. Allow it to grow and become anti-confluent. So what you're going to do is you're going to take out this particular colony and transfer it into a 24 well dish, right? So that it actually should grow, because you know that every cell has a tendency that it is looking for the similar type of cell in the close vicinity. So if you have the 10 centimeter dish, it is actually going to be so big that this colony will be actually not going to get the microenvironment what is required for it to grow, right? Because every cell is looking for some minor microenvironment, some kind of growth signal from the other colony also, other cells actually. So if the plate is very big, this signal is going to be missing. So what you're going to do is you just put a cloning ring on this, you take out this cell and put it into a small well, like the 24 well. So that case, it's actually going to, the concentration of these microenvironment or the growth factors, what it actually secreting is very high, and that's how it's actually going to grow and it becomes confluent. So it's going to grow in large quantity and it's going to be confluent, okay. So allow to grow and become 80%. Transfer these cells to a 6-well dish in the presence of selection media and allow it to grow. So what you're doing is you have taken out from this, you're first putting into 24-well dish, which is actually a small area. Then you're putting it into a 6-well dish, so it's going to be even bigger. And from the 6-well, you can put it into 12-well or even like a large well like this. Okay, so then you can transfer it onto a big dish, and that's why it's actually going to grow. The other method is you can actually be able to use the flow cytometry. Okay, so take a small aliquot of the cell and test the expression of foreign DNA, foreign protein with the help of the Western blotting. In addition, the integration of genome into genome can be checked by external blotting, and you can also be able to do the flow cytometry. As far as the expression is concerned, within the mammalian expression system, you have the two options. You can use the constitutive expression or you can use the inducible expression. Constitutive expression is the place where you are going to use the housekeeping genes, and the protein production is going to be proportional to the cell mass. So higher the cell mass, it is actually going to have the more number of proteins. But the proportion of your protein will be related to the cell growth. So recovery of the protein is going to be a very difficult task when you rely on the constitutive expression. Whereas in the inducible expression system, you can actually be able to use that for the expression of a toxic protein or proteins with the pleiotropic or non-specific effects. In the inducible expression system, you can use the TET on and TET off system. So inducible expression system is useful for the expression of a toxic protein or protein with the pleiotropic or the non-specific effects. The tetracycline controlled inducible system is given. In this system, 7 tandemly related TET operators placed upstream to a minimal CMB promoter and the transcriptional activator gene. In another set, the target gene is replaced with a TTA gene. In the presence of tetracycline, the binding of TTA is blocked to the tetracycline operator. Consequently, it causes the low level of expression of TTA and the target gene. So this is the schematic diagram of explaining how the TET inducible promoter is working. So you have actually TET operators, which actually has the TET, which has the tandemly relayed TET operators, and that is being released, that is being placed upstream to the minimum CMV promoter, and then you have the transcriptional activator gene. So in another set, the target gene is placed with the TTA gene, and in the presence of tetracycline, the binding of the tetracycline to this is actually going to block the tetracycline operator, and consequently it is actually going to induce the expression of the TTA gene and as well as the target gene. So in the absence of tetracycline, the low level of TTA binds to the operator and drive the enhanced expression of TTA, which in turn stimulate the further amplification of the initial signal, and transcriptional activator dropped in the presence, produced in the absence of tetracycline, eventually stimulate the expression of the target gene. So this is all about the expression machinery, what we have discussed for the protein production or the enzyme production. We have discussed about the EKLA expression system. We have discussed about the yeast expression system, insect cell line expression system, and as well as lastly, we have also discussed about the mammalian expression system. Irrespective of the expression system, you can be able to check the overexpression onto the SDS page, but SDS page is only going to say you that there is a overexpression of the particular molecular weight of protein, okay? But it will not going to give you the identity of this protein, and this identity of this protein is very important to know, that a protein of your interest is actually going to be expressed, and that you are actually going to do with the help of a technique which is called as the Western blotting. So let's discuss about the Western blotting. So Western blotting can be done in a multiple step reactions. So what you're going to do is you're going to first so in the Western blotting, what you're going to do is first you're going to run the SDS page. Okay, so you're going to get the pattern of the bands onto the gel, like SDS gel. You're going to transfer that onto a nitrocellulose membrane, and that pattern is going to be maintained, right? And then you are going to treat it with the primary antibody. So primary antibody will go and bind to your target protein, and ultimately you're going to do a washing step. So that washing is going to remove non-specifically bound protein, and it's going to remain allow the specific proteins to be bind, and then you're going to add the secondary antibodies, and secondary antibodies will go and bind to the primary antibody, and then you're going to add the substrate, and that substrate is going to give you the signal. So there are multiple steps what you have to follow, and there is a discrete recipes and protocols what you have to follow to perform the Western blotting. Whatever the material you require. So in this case, we are taking an example of E. coli, which are expressing GFP proteins or GFP protein site. Then you require the protein standard marker, you require the transfer buffer, you require the transfer membrane like the nitrocellulose membrane or the PVDF membrane. You require a plastic tray, spatula, blotting sheets, electroblotting units. You require a reagent for performing the SDS page, that anyway we have discussed in the previous lecture, right? You require the primary antibody, which is the anti-GFP antibody, and you also require the secondary antibodies, and then you also require the developing reagents. So in the step one, you are going to prepare the sample, right? So preparation of the sample depends on the sample type. For the tissue, like for example, solid tissue such as tumor or whole cell or brain, it is first mechanically being broken down into individual cell using the blender, homogenizer or bisonications. Once the individual cells are obtained, it will be processed as described. So first you're going to have the, mechanically you can homogenize and break down the tissue into individual cells. And then these, once the individual cells are incubated with the lysis buffer containing the detergent along with the protease and phosphate buffer cocktail. Then the step two, you're going to do the electrophoresis of the sample, that anyway we have discussed. Samples are resolved onto the SDS page as we discussed previously. Then the step three, you're going to do the transfer of the gel onto the blotting membrane. So that is a very, very important and crucial event, because the how good you are doing the transfer, that actually is going to tell you that is actually going to decide the quality of the Western blot. So first you have to prepare the transfer membrane. So cut the membrane of the same size as gel, and then you are going to have these events. For nitrocellulose membrane, place the membrane in the transfer buffer and observe that the liquids has wicked the membrane. Areas appeared at white spot needs special considerations. Then for the PVDF membrane, immerse the membrane into 100% methanol for 15 to 30 minutes, decant the methanol and submerge the membrane into a transfer buffer for additional 30 minutes. So for the PVDF membrane, you are actually going to have a charging step. Charging step means where you are going to treat the membrane with the 100% methanol, and once you decant the methanol, you can merge the membrane into a transfer buffer for additional 10 to 30 minutes. Okay, so that's how you can be able to make the membrane so that it should be able to bind the proteins. Then in the step 3, you're going to do the assembly of the transfer cassettes. So this is the assembly of the transfer cassettes. You can remove the stacking gel from the page and incubate the gel into the transfer buffer for 10 to 30 minutes. Place a pair of blotting sheets already saturated with the transfer buffer onto the anode plate. Usually it is a black color plate. So first you are going to keep the filter paper onto the anode plates. Then you place the membrane sheets, transfer membrane onto the blotting sheets and remove the trapped air by the rolling test tube or the glass bottles. So first you're going to put the filter papers, then you're going to put the nitrocellulose membrane, then you're going to put the gel, then again you're going to put another sheet of filter paper which is dipped in the transfer buffer, and then you're going to put the cathodes. And after this you're going to tie this up and you're going to put the turn on the electrophoresis. So finally you're going to keep the cathode plates and tighten the transfer cassette by the 4 screws, and after this step you're going to turn on the electrophoresis. So you're going to apply the constant voltage like for 1 hour. So after the transfer, you disassemble the whole assembly and carefully remove the transfer membrane and check the protein transfer by the Ponshu stain. Use a pencil paper and label the different lanes. For example, this is like, so initially since you will going to do a Ponshu stain, you're going to see the bands. So what you're going to do is you take the pencil and you write like this is lane number 1, lane number 2, lane number 3, like this, because this pencil's writing is going to remain there. You cannot use the permanent marker because the subsequent steps probably may remove that. Then in the step 4, you're going to do the blocking. So wash the membrane with distilled water to remove any remaining poncho stain. Put the membrane in blocking buffer containing 5% skim milk. If you're doing the Western blotting for the detecting of phosphorylated protein, then you can use the BSA. Then step 5, you're going to do the probing. So in Western blotting, probing can be done in 2 ways, right? Either you do a 2-step probing or the 1-step probing. In the 2-step probing, membrane is first probed with the primary antibody to recognize the protein of interest, and then membrane is probed with the primary antibody with the approved dilution for 1 hour. Membrane is washed with the buffer containing non-anti-detergent Tritonex 100 and reprobed with another antibody directed against the primary antibody. The secondary antibody is coupled with an enzyme or a fluorescent dye. The washed membrane is incubated with the secondary antibody with an appropriate dilution for 1 hour. Membrane is washed with a buffer containing non-anti-detergent and developed. Use of 2 antibodies increases the sensitivity as well as the giving the flexibility to plan the multiple probes. So if you use the 2 antibodies, first you are going to have the antigen, like that is a protein of your interest, and you are going to first probe that with the primary antibodies, and then you are going to do the washing so that non-specific antibodies are going to remove, and then you are going to put the secondary antibody, and this secondary antibody is actually coupled to some enzyme. So either it can be coupled to HRP or alkaline phosphatase, and this HRP is actually going to give you the signal. Secondary antibody, and why we do so 2-step probing? Because the 2-step probing is actually increasing the sensitivity. In a 1-step probing, you can actually be able to add the primary antibody which contains the enzyme, or you can use the fluorescent table for detection. 1-step probing is not very common. It's a 2-step probing, but is very common. Depending on the enzyme and as well as the different options, you can actually be able to use the multiple ways to develop the blot and detect the protein present onto the membrane. You can use the for example, in the HRP based system, you can use the chromogenic substrate or you can use the luminescence substrate. So, for example, if you want to use the chromogenic substrate, which is actually going to give you the color, you can use the DAB or TMB. For alkaline phosphatase, you can use the BCIP and NBT system. If you want to use the luminescence as the readout, then for the HRP, you can use the luminol or H2O2 system. Whereas for the alkaline phosphatase, you can use the substituted 1,2-dioxane phosphate, and it's actually going to give you dephosphorylated substrate gives the light. Similarly, in the luminol system, the luminol oxidation is going to give you the blue colored light. So after that, you're going to do the colorimetric detections. So wash the membrane with TBS to remove the detergent. Place the membrane into the colorimetric reagent and protein band present in appeared in 10 to 30 minutes. Stop the reaction by washing in distilled buffer. Air dry the membrane and photograph for permanent record. If you want to do a chemiluminescent detections, the chemiluminescent detections are given in the table. Transfer the membrane into the chemiluminescent reagent, soak the membrane for 30 seconds to 5 minutes, drain off the reagent and wrap the membrane into plastic wrap. Place it in a cassette and expose the membrane to a film for a few seconds for 4 hours. Fluorescent detections, you can do the secondary antibody labeled with the fluorescent dye, and that also can be used. And then you can actually be able to capture the signal in a scanner. So to explain all these steps, we have prepared a small demo, and where the students are actually going to show you how you can be able to do the charging of the membrane, how you can be able to assemble the cassettes to transfer the bands from the SDS page to the nitrocellulose membrane, and so on.
Hi everyone, myself Swayam Prabha, Research Scholar at Department of Biosciences and Bioengineering, IIT Bombay. In this video, we will demonstrate you how to do a Western blot and how to analyze the result using ECL, electrochemiluminescence substrate. So here what we will do, we have to run gel first, then we will transfer. The transfer method, how to do the transfer, we will show in this video. In previous video we have already shown that how to prepare a SDS pager and how to run protein samples. So in this video particularly we are interested in factors associated with the Western blotting. For doing Western blot, we need membrane and transfer buffer and the transfer medium. This one is we use to transfer this gel to membrane. So here membrane can be two kind. One is nitrocellulose, which has low protein binding efficiency and hydrophilic in nature. Another membrane is PVDF. This is hydrophobic membrane and higher protein binding capacity. So the processing for Western blot is different for nitrocellulose and PVDF. If you are using PVDF membrane, we have to cut the part. Either if you have ready-made pre-cut blots, then no need. If you are taking from a bundle, you have to cut precisely how many wells you want. So after that, you have to label front on the blot, where that front side can be used for transferring the protein, and that can be used in previous step, further steps also, like antibody incubation. So here for if you want to use PVDF membrane, you have to charge with the methanol. So since the PVDF is a hydrophobic membrane, you cannot directly transfer in the aqueous medium. First you have to keep in methanol for at least 20 minutes. So after this can be called as charging. So after this we will use that for transfer. So this is pre-soaked in methanol and equilibrated in transfer buffer. So here while doing uh, transfer, we need to consider few things. The buffer always should be in chilled condition, otherwise during this transfer at high voltage it will generate high temperatures, so that may degrade your protein or decrease the efficiency of the transfer. That's why we need to keep the buffer always in chilled condition. And let's start the procedure. So we need a pre-run gel. So we already finished the gel running. In addition to that, we also need sponges which will give cushion to the gel, so that gel may not destroyed during the transfer. So this is the cassette we'll use for the transfer. So this is negative side of cassette and this is the positive side. So we are going to keep gel on negative side and positive side the blood membrane. So when we apply voltage from this side to this side, the negative protein, it will be transferred, it will be moved to positive side and it will be captured in the membrane. So first, for doing that, these sponges we need to keep, and also this maybe give some non-specific binding to membrane. So what we will do, we will put blotting sheets on top of this. So after this, you have to remove air bubbles if any present. So once you inserted the blotting sheet, then you have to keep your gel. So here we have to remember that gel, after finishing the SDS phase running, you have to keep in transfer buffer, so that it will give identical condition or equilibration kind of thing during transfer, so that protein transfer may be easy. So this is the gel and keeping on the negative side. So after that, we have to overlay with the membrane. Next, we have to remove any air bubbles if present. We have to overlay with another blotting sheet and remove the air bubbles. Each and every time when you introduce something, you have to remove air bubbles. So this is the final sheet. So this is the positive side of the cassette. Just have to keep like this. These are the screws. We have to tighten it up. Then only the contact between the gel and the membrane will be sufficient to get transferred. First, you don't tight initially. You just keep, and after that, press the positive side of the cassette, then tight the screws. So all these things should be done in the transfer buffer only, unless specified. So this is the chilled transfer buffer. Now we are going to do transfer. Pour sufficient buffer keep this ice pack also. If the chilling is not sufficient, then there may be heat generation. So in order to prevent that, we will use this ice pack. So this will keep the buffer cool till the transfer, end of the transfer. So once that is over, you directly take out the cassette and keep. If there is a buffer insufficiency, you can add on top of that. Make sure that the cassette completely submerged, so that the transfer will be proper and there is no air bubbles. So once the setup is over, now you can transfer. Now transfer is going on. So how much voltage we need to give, it depends on transfer to transfer, it varies. Generally in our lab we will give at least 2 hours of transfer at 120 volts, which is sufficient to transfer even low molecular weight proteins also. But from instrument to instrument also it varies. You needed to optimize before doing transfer. After 2 hours, we have to remove the blood and incubate with the blocking buffer. So I am going to stop here. Remove the cassette, keep it in a tray. Remove the screws properly. Gently remove the sponges. Take out the blood and keep it in blocking buffer. In this condition, we have to keep, if you are keeping it room temperature, it is for 2 hours at least. If you are keeping in 4 degrees Celsius, you can keep overnight. The blocking buffer contains skim milk or BSA along with the Queen 20. The western blot transfer, it's all depends on the efficiency, how precisely you are doing the transfer. For example, you should not use your bare hands while handling the blot or gel. So whatever the proteins present on your fingers, it will transfer into gel or membrane, which will give high background during development of the blot. So always use gloves. Apart from that, while handling the instrument, make sure that there may be possibility of electricity, the shock may happen sometime. So we have to, that time also we need to use gloves. And after finishing the transfer, you have to clean all the apparatus properly and dry it for the next time use. After the blocking of the membrane, we have to remove the membrane and incubate with the primary antibody without washing. The main purpose of the blocking is that it will occupy non-specific sites other than the respective protein, so that when antibody comes, it will bind to that specific protein and gives no noise. So after this, we will incubate with the primary antibody for overnight at 4 degrees Celsius, then wash 3 times, at least 15 minutes each, with the TBST buffer or PBST buffer, and again incubate with the secondary antibody, suitable secondary antibody, for 5 hours at 4 degrees Celsius or 2 hours at 3 hours at room temperature. After that, we need to wash properly at least 3 times. Then we will develop the blood with the electrochemiluminescent substrate. In earlier Western blot, how to do Western blot video, we showed how to transfer the proteins to membrane. So we incubated with the primary antibody, following secondary antibody, and washed thoroughly. Now here we show how to develop a blot. For developing a blot, we need chemiluminescent substrate. In most of the commercially available kits, luminal is the one of the substrate we use it for this purpose. So luminal in presence of hydrogen peroxide and peroxidase enzyme, which present in the secondary antibody, horse radish peroxidase conjugated secondary antibody. This horse radish peroxidase converts luminal to excited state luminal by deprotonating and oxidizing it. So this product, this excited state product gradually leaves the energy by releasing luminescent photons. That light will be detected using this instrument. So these are the commercially available chemiluminescent substrate solutions. So it is available from a wide range of companies. We have to mix 1:1 ratio. So we have to take out the blot, drain the buffer whatever present properly, so after that you keep blot in between a plastic paper foils. Then we will take chemiluminescent substrate, so after that you have to slowly press and remove air bubbles. This is the tray we use it for developing the blot. So we have to open the system, properly align the tray, and then shift blot to the tray. Once it is over, you have to just close. Here we have to select application. We want blots, that is chemiluminescent, and what exposure want. You have two options, manual, auto. In auto two options are there, optimal auto exposure, rapid auto exposure. We will choose optimal auto exposure. So you can enlarge the blot also. Once it is over, you just say so this is the developed blot. So as we can see the bands pattern. So this is how we develop Western blot through electrochemiluminescent substrate. So in this video we demonstrated how to transfer proteins to a blot and what are the precautions need to be taken while doing the Western blot, and also how to develop the blot and what is the laying principle behind the developing the blot. So I hope this will help you to understand the basic outline mechanism of how Western blot works. Thanks for watching.
So in the demo, students have explained the different steps, and I hope the demo video could be helpful for you to understand the practical aspect of the same Western blotting. and it will help you to perform the experiment in your laboratory. So what we have discussed about the expression of the protein utilizing the mammalian expression system. And in that you can have the two options, either you can have the transient expression system or the permanent expression system. And both of these options have their own positives and negatives. And apart from that, you can also have the option of non-constitutive expression or the inducible expression system. And so based on the different types of requirements and as well as the criteria, you can have the choice of choosing the different conditions for expressing the protein into the mammalian expression system. And once you are going to express the protein of your interest, you can also should perform the specific test like the Western blotting to detect the protein on the nitrocellulose membrane. So with this, I would like to conclude my lecture here. In our subsequent lectures, we are going to discuss about the protein purifications, so that you can be able to isolate the enzyme of your interest from the crude lysate. Thank you.